Well, I think I'll get started. Looks like it's about 8. I think probably most of you know who I am. This is actually sort of my silver anniversary address. I've been here for 25 years. And so it's going to be kind of a stroll down memory lane. Sort of. Some of the highlights and perhaps lowlights of the last 25 years. But one of the things that I've become very interested in is degenerative ataxia, and I'll sort of tell you how that came about as we go through the talk. First, I have some disclosures. I have no financial relationships, and I will not discuss any off-label use of drugs. So what I want to do today is sort of go through the basic anatomy of the cerebellar system, because this is the system that falls apart in ataxia. We'll talk a little bit about the pathology that leads to ataxia, and then talk quite a bit about animal models that we've used to try to understand pathogenesis of ataxia, but also talk a bit about what we've done with clinical examinations, autopsy examinations of patients with various types of ataxia. So we'll start off just sort of telling you what ataxia is, and then I'll talk about the anatomy and the pathology, introduction to ataxia models, a couple of models that we've used, and then talk about several human forms of ataxia that we've dealt with over the years. So what is ataxia? It's basically a disease of the nervous system that results in incoordination. So velocity, amplitude, direction, timing of movement are thrown off. And the clinical features are usually problems with gait, problems with measured movement, so people don't stop movements at the right point. Rapid alternating movements are affected. Speech is often affected, dysarthric speech, problems with eye movements. And basically, this is what happens if you become intoxicated with alcohol. So the cerebellar system is affected quite frequently, and actually highway patrolmen are very good at doing the cerebellar examination, because that's so which parts of the brain are involved? So we usually think of the cerebellum as being the major player here, and that's probably true, but there's a great deal of other inputs. So the pons, the medulla, particularly one aspect of the medulla, and even the cerebral cortex plays a role in the overall integration, as does the spinal cord. So the medulla, there's a structure called the inferior olivary nucleus that's very important and has large projections that go to the cerebellar cortex on the opposite side. So this is a structure that's very commonly affected in various forms of ataxia. The pons, which is in front of the medulla, is located here. The cortical input goes through the pons, so the cerebral cortical input goes through the pons, and the pons, again, the connections cross over to the opposite hemisphere. There are large numbers of cell bodies here, and they're one of the major driving forces in the cerebellar cortex. So the cerebellar cortex is sort of the star of our show here, and particularly the Purkinje cell, which is sort of the prima donna of all the neurons. These are big cells that have large dendritic trees, as shown by this Calbinin staining, that extend into this upper layer, which is known as the molecular layer. These cells are driven predominantly by these tiny little neurons here. Basically, all you see are their nuclei. These are called the granular neurons, and these give rise to excitatory fibers that stimulate the Purkinje cells in the dendritic, or in their dendritic fields in the molecular layer. These are excitatory amino acid transmitters, so they're glutamate. And glutamate can sometimes be toxic to cells if it's not properly regulated. There are a bunch of small neurons in the molecular layer that are actually inhibitory neurons. They're GABAergic neurons that sort of, they also are stimulated by these cells, but they modulate the activity of the Purkinje cells. Some of these deeper cells in the molecular layer send little fibers to the cell bodies of the neurons, and these are known as basket cells, and the fibers are called basket fibers. And I'll show you what that looks like in a few minutes. So the cerebellar system in general, there's inputs to it, there's the cerebellum itself, and then there's the outputs of the cerebellum. So the inputs come from the spinal cord and cranial nerves. So this tells you where your body is in space. Particularly the vestibular nerve is involved with balance. If you want to initiate movement, there are inputs from the cerebral cortex that go via the pons. And then the olivary nuclei are particularly involved in sort of the oscillatory patterns that are important for cerebellar function. And they get connections from the deep cerebellar nuclei, sort of in an indirect fashion. The cerebellar cortex itself, we've already talked about, so the organization of that. The Purkinje cells are the only output of the cerebellar cortex, and their fibers go to the deep nuclei, and then the deep nuclei project to other parts of the brain, mostly to the thalamus, which then projects to the cerebral cortex and to other sites. So it's a fairly complex system, and this is sort of a schematic showing sort of the primary wiring. You have Purkinje cells in the cerebellar cortex, you have the granular cells. The granular cells have what are called parallel fibers that run perpendicular to the Purkinje cell dendrites. Their input is predominantly from the pons and from the spinal cord and from the vestibular system. So indirectly, those areas affect the Purkinje cells. They don't have any direct connections. The only direct connection from outside the cerebellum is from the inferior olive. And they have what are known as climbing fibers that actually have a lot of veto power onto whether or not cells fire because they're very close to the primary dendrite and so have much more influence than these parallel fibers have. The output then goes to the deep nuclei, and the deep nuclei project to other sites. This is going to be on the test. So this is the whole wiring diagram. And the whole point of this is not to remember this, but that if any part of the system goes haywire, the end result is ataxia. So there are many ways to get to the same place. So what we typically see in ataxias are various parts of this system falling apart. And one of the most common, of course, is cortical atrophy. So you have loss of Purkinje cells, usually to much greater extent than loss of granular cells. In a number of them, the deep nuclei are also affected. And in many, olivary atrophy is a component of it. One thing about the olive is that it's the major input to Purkinje cells, really the only major target for them. And if the Purkinje cells drop out in great numbers, the olivary neurons sometimes die secondarily because they have no reason to live. They lose the trophic influences from their targets. A number of forms of ataxia also have degeneration of the basal pons, which is the part that projects to the cerebellum. And then some other ones have predominantly spinal cord atrophy, like Friedreich's ataxia. Has a lot of spinal cord problems. And then of major importance, for particularly the autosomal dominant ataxias, is that there's often involvement of systems outside the cerebellar system as well. So you can have motor neuron disease, you can have problems with the extrapyramidal motor system, you can have problems with peripheral nerve and even cerebral cortex. And some of these changes are actually the most life-threatening aspects. So many of the ataxias are not necessarily life-shortening, but they can make life difficult because of inability to ambulate properly and communicate properly. So what we see often is cortical degeneration, and often it involves the anterior and superior parts of the cerebellum more fiercely than other parts. I'm not quite sure why that is, but we see it fairly frequently. And so what you see is, on the left is normal cerebellar cortex. You see the regularity and number of Purkinje cells. This is a little bit higher power, but you can see that in this field there's one Purkinje cell. And this is a patient with a form of cerebellar degeneration known as SCA6. Another feature that's quite common with cerebellar disease is to see these dilations of the proximal axons of the Purkinje cells. And these are known as torpedoes, as you can probably guess from their shape. They typically arise in the proximal part of the axonal process in the granular layer. And they're actually not interruptions, they're actually just sort of like constipation of axonal transport. As the material gets hung up there, but you can see the fiber goes on past them. It's sort of a stereotypic response, and it's seen in many types of ataxia, including hereditary ones, alcoholic ataxia, all sorts of forms. These are the basket fibers I was talking about. So here's a Purkinje cell that's sort of bathed in these fibers that surround the cell body, and when the Purkinje cells degenerate, you often see what are known as empty baskets. So the basket fibers are still there, but the cell that was innervated is no longer present. Some patients have severe atrophy of the pons. So here we can see, this is the normal appearance of the pons. This is a myelin stain. And you can see this is a patient with severe olivopontocerebellar degeneration. The only fibers that are going through here are the descending motor fibers. So these are fibers that are running sort of north and south. And all the ones that are running east and west are basically missing. And this is actually the fifth nerve coming through here. So that remains intact. But the rest of this is almost completely degenerated. This is an example of olivary atrophy. The olives usually kind of bulge out laterally around the edge of the medulla, and then the pyramids. This is the corticospinal tract. Normally are sort of partially surrounded by the olive, and here they're kind of dangling down from below. So there are some pathological classifications of ataxia that are semi-useful, but they're not really all that informative in many respects. So one pattern is this cerebellar cortical degeneration. This is probably the most common one, where you basically see primarily Purkinje cell loss and maybe some loss of granular cells and some of the interneurons. And they may have secondary atrophy of the olives. Olivopontocerebellar degeneration is one that's quite striking, where there's sort of what I showed you previously with a really severe atrophy of the basal pons and atrophy of the olives, along with cortical atrophy of the cerebellum. And in a number of conditions, particularly some of the hereditary spinocerebellar ataxias, that's also accompanied by involvement of other parts of the brain that may or may not be related to the cerebellar system. And then there are some that are predominantly spinal and deep nuclear, so like Friedreich's ataxia, where there's loss of afferent projections to the cerebellum, and the deep nucleus is affected, but the cerebellar cortex looks almost completely normal. So you can get to the same place with different types of pathology. So one of the historical problems with ataxia was we tried to classify them by pathology, but there was really extensive overlap, and so different diseases all kind of look the same. And the other problem was that people who had hereditary ataxias, they didn't all look the same. So there was variation within the same family. If you classified by the pathology, you might not even put them in the same group, which didn't make any sense. Clinically, sort of the same thing. There was a lot of lumping, because, I mean, if the system falls apart, the end result is usually ataxia. So there's some subtle differences that may be unique to certain types, but for the most part, they look quite a bit alike. So one of the major things that's happened in the field of ataxia since in the last 25 years has been the genetics. So in 1990, we didn't know anything about any disease. We had no genes identified. So it's really been a major breakthrough in terms of understanding classification, or at least having something to hang your hat on. And if we know what the genetic defect, then you can start to look at molecular mechanisms to try to figure out what's going on. Primary degenerative ataxias, so there are inherited ones, autosomal recessive, autosomal dominant. I'm going to talk almost exclusively today about dominant ones. There are some X-linked ones and there's some sporadic ones. So of the hereditary ones, the recessive ones, the most important in North America is Friedreich's ataxia, which is reasonably common, but I won't be dealing with that today. The dominantly inherited ones, the ones I'm going to be talking about today are the ones that have, the motif is that they have a polyglutamine repeat within the affected gene. And I'll get into that in more detail in a little bit. But there are now up to SCA37. So there's been a lot of work done over the last 20 years or so. So my introduction to ataxia models started almost the day I walked in the door. I was down in the basement of Deal Hall, and Harry Orr wandered down and said, "You know, I've got these really weird mice." And what he was trying to do was to immortalize Purkinje cells, and so he could grow them in culture, things like that. So the strategy was to try to transform them with SV40T antigen. So using a promoter that they had developed called the PCP2 promoter that expressed exclusively in Purkinje cells, they were going to try to immortalize them with the SV40T antigen. what happened was that they had these mice that got very ataxic, often very early in life, and when we looked at them, they didn't have much of a cerebellum at all. And then there were some other mice that used the same strategy where apparently the gene turned on a little later. They developed normally and then over the period of a couple months became quite ataxic. So this generated some rather interesting work that this was in the early 1990s. There was a postdoc named Rod Feddersen who was working in Harry's lab at the time who did much of this work. So this is what the one line looked like, the ones that had very early onset ataxia. So this is what a normal mouse looks like at 3 days postnatal. So the cerebellum is kind of interesting in rodents, that it's almost entirely a postnatal event, the formation of the cerebellum. So they have a 21-day gestation period, and in about 21 days postnatally, the cerebellum develops, and it's pretty much completed by 21 days. And one of the things about cerebellar development is that the granular cells actually start on the outside of the brain, and they migrate into the internal granular layer. So you can see that there's an extra layer here at 3 days. This is what the SV5s look like at 3 days. This is kind of a little stub of cerebellum. This is what the normal cerebellum would look like after maturation in a mouse. And this is what the cerebellum looked like in the SV5. And this is actually blown up, so this is not in scale, so this is actually much, much smaller than this. The other line, the SV4 line, where they started off relatively normally. They had a lot of Purkinje cells in the normal place right off the bat, so this is day 1 postnatal. By day 32, however, they were becoming much more sparse. There's only 2 or 3 of them in this field, and this is the normal appearance where they're really stretched all the way across. And by day 70, when they had cage behavior of ataxia, they were quite depleted, more than 50% loss of cells. Rod was interested sort of in the mechanism of this, and so one of the things that was known was that T antigen binds both the P53 as well as the retinoblastoma PRB, the retinoblastoma gene, the tumor suppressor gene. And so he played around with mutating the T antigen in different ways so that either it bound to one or the other. And he got quite different phenotypes when he did that. So when you eliminated the P53 site but left the PRB site intact, it looked quite a bit like the SV5 models. So there was a very early loss of Purkinje cells. When they eliminated the retinoblastoma gene, or retinoblastoma protein binding site, then was kind of an interesting pattern. The animals became very ataxic, but they didn't really lose Purkinje cells, but the Purkinje cells didn't develop completely normally. So there was like arrested development in a sense. I'll show you what that looks like in a sec. So one of the things that T antigen does is it actually binds the retinoblastoma protein, which releases a transcription factor called E2F, which tends to send cells into cell cycle. So that's how it sort of is tumorigenic in the typical setting where T antigen integrates into the normal genome. In the Purkinje cells, however, by the time the PCP2 gene turned on to express the transgenic T antigen, they were already committed to differentiation. And so they would begin to try to go through cell cycle, and they would actually incorporate BRDU. But rather than dividing, they would undergo an apoptotic pathway, and we showed this by nick end labeling kind of approach to show that there was actually an apoptotic type cell death. So this is one of those lines. This is the one with only the PRB binding. So this is at 9 days. It looks pretty good. There's a few areas where maybe there's a little bit of loss of Purkinje cells here. By 15 days, it's starting to look a little more ragged, and by 1 month, it actually is quite atrophic. And we were able to catch some of these cells in the process of cell death and showed that they were actually undergoing apoptosis. So this is kind of the scheme. They first turn on the T antigen, they start to incorporate BRDU, and then they have the nuclear fragmentation and cell death. And all this happens over a fairly short period of time. The animals with the P53 binding that was intact, so this is what the normal appearance of the cerebellum. This is again Kalbindin staining, which shows the dendritic tree of the Purkinje cells very nicely. This is at scale, so these are the animals whose Purkinje cells are quite stunted. They're all there, but they're only about half the size they should be. And these animals are ataxic very early as well. So it's not just cell loss that can cause ataxia. So if the connections are not properly made or not properly maintained, you can still have an ataxic state. So some of the lessons we learned from this model were that certainly you can get ataxia by killing Purkinje cells, but it takes quite a few. So even in that late onset animal model, they had to lose more than 50% of their cells before they actually became ataxic. So there's a lot of redundancy that's built into the system. We also then showed that abnormalities of the sort of the anatomy of the Purkinje cells can be just as detrimental to function as having lost half your cells. But probably the most important thing that we learned from this was that this strategy could be used for other genes that might be important. And this was at the time when Harry's lab in conjunction with Huda Zogby's lab at Baylor were actually found the gene for SCA1 and showed that it was a trinucleotide repeat expansion in a protein known as ataxin-1. We'll get into that in a minute. But we now have a way that you can introduce a gene into a cell that's very important for the function of the cerebellum, so that if that cell is dysfunctional or drops out, you have not only a pathological readout, but you have a functional readout. They become clinically ataxic. So I just want to give you a brief introduction to some of the dominantly inherited ataxias as we get into the SCA1 model. So there are, as I mentioned, there are now 37 numbered, although some of them, there's some numbers that are skipped. And it turns out that a couple of them are probably the same thing, so there's probably not quite 37. But there will be more, I'm sure. But one of the common motifs, particularly in some of the more frequent occurring SCAs, are the fact that they have this polyglutamine expansion. So these proteins typically have an area within the translated area of the protein of multiple glutamine residues. But typically there are fewer than 35 to 40. And sometimes they're interrupted by histidine. But all of these genes, or all of these diseases have, the theme is that they have an expansion of that stretch within the protein. And once it gets above roughly 40 amino acids, it becomes pathogenic. Each disease has a different gene, so the mechanism is similar, but they're all in a different protein. And many of them also they develop aggregates in the nuclei that are formed from the mutant protein that is also ubiquitinated, so you can identify them either with ubiquitin antibodies or with antibodies against a specific protein. And examples of this polyglutamine family are, of course, Huntington's disease, Kennedy's disease. So Huntington's is a disease that primarily affects the basal ganglia. Kennedy's disease is a lower motor neuron disease, but all the rest of these are ataxias. So there's something about this that seems to affect the motor components of the brain somewhat selectively. This is an example of what these inclusions look like. So this is a section from the pons in a patient with SCA3. You don't see these by H&E, but they miraculously appear with ubiquitin stains or a stain against ataxin-3. This is just a list of the ones that cause ataxia. Most of them, the function of the protein is not known. The one exception here, the major exception, is SCA6, where the protein is actually a calcium channel. And we'll return to that a little bit later in the talk. So SCA1 is a autosomal dominant ataxia, onset usually in midlife. The duration is typically 10 to 15 years after onset. It's a fairly severe disease, and patients die largely from problems that are outside the cerebellar system, where they have cranial nerve nuclear involvement and develop problems, which is very much like you might see with ALS. Pathologically, it can be quite variable, and that seems to be somewhat depending on the size of the repeat expansion. So the general rule is that the longer the repeat, the earlier the onset and the more severe the course. So people with short repeats tend to have later onset disease that's milder. People that have longer repeats tend to have more severe disease. Most patients have cerebellar cortical degeneration as well as an olivopontocerebellar degeneration. So they lose the basal Pontine neurons as well. The deep nuclei are typically affected as well. So it's like a quadruple whammy. Like every part of the system is falling apart. So it's not mysterious as to why these people really have problems. But the thing that's probably the most important is the lower motor neuron disease, because that's what tends to kill these patients. The gene is on chromosome 6. The gene product is known as ataxin 1. It's roughly 800 amino acid, depending upon how long the CAG repeat is. So that's somewhat variable from person to person. Even in this room, people have quite a range. It's expressed in many cell types, not just in neurons, but it has a nuclear localization in neurons. Its function is still somewhat unknown. We'll get into that in a little bit. And we do know that you can knock this protein out, and the animals do not have a neurological phenotype, which is very handy for us because now we can cross mice to the knockout background and look at the effects of the transgene almost exclusively. The mutation is due, as I mentioned, to the expansion of that polyglutamine tract. Typically, there's loss of the histidine interruptions and then expansion up to around 40 or more repeats. It's thought to be basically a gain of function mechanism rather than a loss of function. Pathologically, you see, again, atrophy of Purkinje cells, eventually Purkinje cell loss, and ubiquitinated inclusions are present in many cell types. Interestingly, not present in Purkinje cells in the native disease. So Harry's lab, Eric Burright, who was a postdoc in Harry's lab, developed, using the same strategy as the SV40 mice, developed a mouse where we introduced the mutant SCA1 gene so that would express in Purkinje cells. So it's not a model of the human disease completely, but it's a very good way to look at sort of the mechanisms of disease. And you have that phenotypic readout. So you have clinical ataxia as well as pathology that you can look at. And the Purkinje cells are very nice because they have a very well-defined dendrite tree, and when it's abnormal, you can usually see that pretty easily using Kalbina staining or other methods. These animals got cage behavior ataxia by about 3 months of age, and it actually had subtle changes of coordination even earlier than that. The cerebellum appeared to develop normally, so this is what it looked like. So at 25 days, it actually looks quite normal. Even by 6 weeks, it looks pretty normal, but at high power, sometimes you could see some pruning of the dendrites very close to the cell body. By 15 weeks, where they already have ataxia by cage behavior, you can see now they're becoming quite truncated. There's a lot of loss of branching in the proximal aspects, and some of the cells are starting to drift upward into the molecular layer. Interestingly, here you can see, this is at about 6 months, you can see that there's a few cells that for some reason didn't turn the gene on, so they're normal. So you see these really nice looking normal Purkinje cells, and these are their little stunted cousins who are very abnormal. But the cell loss is relatively sparse at this stage. So these animals are severely ataxic, but not so much from loss of Purkinje cells, but just from their anatomical changes. If you take one of the important control was if you look at if you put the normal human gene and overexpress it into Purkinje cells. So we just took a 30 repeat instead of, we used an 82 repeat for the disease model. Those animals did not get sick, and their cerebellum, with a few little exceptions, they had a little occasional torpedoes, but phenotypically they did not have any kind of a movement disorder, and their Purkinje cells actually looked quite intact. So it wasn't just overexpression of protein, it was overexpression of the mutant protein that caused the problem. So using this same sort of scheme, Harry's lab, over a period of a few years, actually did some very important experiments that led to some insight into the underlying mechanisms here. So this is just showing what I've already showed you. These are the controls with the 30 repeats, and then the affected with 82 repeats, and you get not only no pathology here, but the distribution of the ataxin in the nucleus is very widely, diffusely distributed as it would be normally. In the animals with the abnormal protein, the ataxin tended to aggregate into these clusters. There was a MD PhD student in Harry's lab named Ivan Clement who mutated the nuclear localization sequence. So they overexpressed the protein in Purkinje cells, but it wasn't able to get into the nucleus. So you see there's a lot of cytoplasmic protein, and the animals did not get disease. So the protein has to get into the nucleus in order to cause disease. Another important experiment was done by Pam Skinner, who was in the lab at about the same time, in which they deleted the self-association region, because there was a lot of controversy about whether these inclusions were important in the pathogenesis of the disease. So when she did that, the protein was widely distributed, just like it was in these animals, but the animals got sick. So the aggregation of the protein didn't seem to be really the important aspect, but the protein was in the nucleus where it could cause damage. Another sort of a post-translational modification that was important was phosphorylation of ataxin. And Harry's lab found that one of the important sites of phosphorylation was a particular serine residue right next to the nuclear localization sequence. And so they decided to try to mutate that site. So, fairly interesting system. So alanine and serine have the same backbone structure, but serine has a hydroxyl group that can be phosphorylated. So they substituted serine for an alanine, and those animals really did not get sick. So this is the model I've already shown you. So at 18 weeks, you can see there's already considerable Purkinje cell degeneration. At 27 weeks, even worse, and 37 weeks, even worse. But in these animals where they substituted the alanine for the serine, you can see that the Purkinje cells are much more robust. There's probably some pathology that occurs over time, but it's much, much slower, even though they're expressed at equal levels. So another critical experiment was done about 10 years ago now. Tao Zhu, who was working in Harry's lab, was involved in this. And this was sort of a proof of principle in terms of, could you affect the disease by turning the gene off? So if you stop making the mutant protein, would that improve the phenotype? So this was a so-called TET responsive, so it's under the control of the tetracycline responsive element. In this model, it was TET off, so if you gave the animals doxycycline, the gene would turn off. So first of all, the animal model that was developed in the absence of tetracycline looked very much like our classic model. So the pathology was really quite similar to the animals we developed up to now. So they developed pretty much the same kind of pathology in roughly the same time course. So the way we evaluate ataxia in the lab clinically is via a rotarod. So this is sort of a log rolling contest kind of thing where the animals have to get on a spinning dowel, and we time how long it takes, how long they stay on. It's not the perfect test because motivation can be a major factor, and other motor abnormalities can come into play as well, so weakness, things like that. So it's not exclusively a cerebellar test. You can see that these two guys on the left have failed miserably, but I've protected their identity. So the way this is done is you do the trials over a 4-day period, and there's somewhat of a learning curve that's involved. So nobody's very good at it the first day, but they start to get better, and you see some separation over time. So this is actually just looking at wild-type animals versus the transgenic. So the dotted line is the classical transgenic that I've talked about already. The dashed line is the one that's TET responsive, and their slopes are pretty similar. So they basically behave the same at early times at 12 weeks and at 24 weeks. If you give the animals doxycycline 6 weeks into life, so the abnormal protein's been there during development, and for this first 6 weeks after birth. Then you turn the gene off and wait 6 weeks. So this is what the animal who's had the gene on the whole time looks like. This is what the wild type looks like, and this is what the animal who had the gene turned off. So you get an improvement almost back to normal if you turn the gene off very early. What do you see pathologically? So this is what it looks like at 6 weeks on. This is what it looks like at 12 weeks on. And this is what it looks like if you turn the gene off halfway. So you get considerable improvement, actually even over what it looked like at 6 weeks on. So you see much more robust and full dendritic tree. If you wait 12 weeks before you turn the gene off, you see a more modest phenotypic response or behavioral response. So here you can see this is just 12 weeks on. You turn the gene off for 4 weeks, you really don't see improvement. You turn the gene off for 8 weeks or 12 weeks, you see some improvement. So these lines are essentially superimposed. They're not as bad as the ones that have the gene on for the full time, but they're not as good as the animals who are normal. Pathologically, what you see, again, this is what 16 weeks on looks like. Maybe some improvement if you've taken it off for 4 weeks. It looks a little better after 8 weeks off, and looks even better after 12 weeks off. We also tried doing this later with the animals starting at like 24 weeks. We got much more modest response. So sort of the bottom line is that you can turn it off, but the earlier you turn it off, the better the recovery. And if you wait too long, you only get some modest recovery. We weren't really able to see much change in the Rotarod behavior when they were turned off late, but there was some change in cage behavior. And the Purkinje cells looked a little better, but not really particularly great. So, sort of the conclusions from this study is that no matter that if you remove the newly produced mutant protein, it seems to improve the phenotype. So that would suggest that the turnover of the protein is relatively rapid, and that this is really stage dependent. So the earlier you do it, the more likely you are to get a favorable outcome. So this is kind of a proof of principle. So if you could find a way to knock down the protein in some way with genetic manipulation, this might be a strategy. So what Harry's lab is doing now, they're looking at inhibitory RNAs that you could use viral vectors. Delivery is going to be somewhat of an issue. It works fairly well with Purkinje cells because you can inject into the deep nuclei, and you get, because the axons from the Purkinje cells go to the deep nuclei, you get pretty good transfection of the Purkinje cells. But it's going to be a much more difficult situation in the human disease, I think. But again, we can use both structure and function as markers of therapeutic benefit. So for disease models, I think it's an important proof of principle. So just kind of to sum up the investigations in SCA1, transgenic models have really been very important in sort of understanding to date. And there's a lot more I haven't talked about, particularly over the last 10 years, that I'm not dealing with today. But an important concept was nuclear localization of the mutant ataxian. Post-translational modifications to phosphorylation is very important. And that is important in the sense that it may be another targetable aspect of the disease. So if you could in some way prevent the phosphorylation. So Harry's lab has been looking at that. It's a kind of daunting because often these kinases are very ubiquitous, and it might be a kinase you need for something else. So knocking it down completely may be detrimental to the patient. But we really don't know what the mechanism of action is yet. We know that the ataxin 1 protein is associated with many nuclear factors that are important for transcription, and Harry's group has done a lot of work in gene expression analyses, comparing these mice to wild-type mice and whatnot. And so there's many alterations of gene expression. And really, what are the most important ones is something we're still sort of grappling with. So I want to kind of change gears a little bit and talk more about studies that we've done with human disease. So SCA2 is another trinucleotide repeat condition. It's probably more common than SCA1 actually. And pathologically it looks a lot, the classical cases look a lot like SCA1. They have oligopontocerebellar degeneration, a little bit less involvement of the deep nuclei, and some of the brainstem involvement may be a little different than you see in SCA1. One of the differences, though, is that in some patients with SCA2, they have massive repeat expansions. So in SCA1, the largest repeat expansion ever reported in a human patient was like 83. So that's why we used 82 as the sort of to try to make it as fierce as possible. And the patient with 82 repeats or 83 repeats actually had a funny looking disease. She was a kid who developed ataxia at age 4, died at age 10, had no Pontine involvement whatsoever. So it did not look like classical SCA1. But these kids with the massive expansions in SCA2 really look different. They have an infantile onset. Some of them have almost no cerebellar involvement at all. Some of them have some. And this phenotype, due to excessive expansion, can also be seen in Huntington's disease where they get early onset and the disease looks somewhat different. They tend to get a Parkinsonian kind of picture clinically, and the pathology progresses much more quickly. SCA7 is another one that can have infantile onset. So this is what the pons looks like in classical SCA2. There's sort of a loss of the volume of the pons. You can see that it's much narrower, has almost kind of a keel-like shape to it because of the severe atrophy. These kids who have the massive expansions have a range of pathology. Some of them have quite a bit of cerebellar degeneration, but the pons in many of them looks completely normal. So you can see this anterior bulge, which is quite normal. Some of them have very severe white matter disease. This is a flare image, so the white matter should be dark here, not white. And so it's just basically a complete loss of the myelin in the so it's like a leukodystrophy essentially. So phenotypically looks quite different. We were able to study a couple of these patients at autopsy, and quite interestingly, there was one patient who had like 500 repeats. Sometimes it's hard to actually measure them completely accurately, I think, because of the methodology, but had really massive expansion. And the cerebellum looked almost normal. You can see that there are large numbers of Purkinje cells. The granular layer looks nice, molecular layer looks nice. But another patient who had 300 repeats, this is shot at the same magnification. You can see the cerebellar cortex is down to like 1/3 or 1/4 the size of this. Purkinje cells are all gone. Granular cells are basically all gone. So a really quite diverse pathology. So why is that important? Well, one of the things that we do in our model systems is we try to accelerate disease because you don't want to wait 2 years for a mouse to get sick. I mean, it's hard to do experiments that way. So there's a couple ways you can jack that up. You can increase the expression by putting multiple copies of the gene in, which is what Harry's group has done. And you can also expand or make the repeat size bigger because you'll make it a more rapidly progressive course. And that's what Harry's group also did. So they used kind of both strategies. So we used the highest number ever shown in a human patient as well as overexpressing it in Purkinje cells. Hudazagbi's lab has tried to make a more true-to-life model of the disease, and using, again, like a 78 repeat, got an animal that really had almost no phenotype throughout life. They really didn't look very sick. Of course, that's under the expression, basically it's a single gene with it's being run by its own promoter, so it's not being driven by anything. So she tried to make it worse by expanding the repeat size, and so with 154 repeats, she got animals that got very sick, but they don't look like SCA1. They don't have cerebellar, very little cerebellar degeneration. They die by the time they're about 6 months old. Their brains are very, very small, but it really doesn't look like SCA1. The other thing is that because of this phenotypic variation, targeting specific cell types may be very difficult to do because we don't know which cell types are going to be involved in every patient. So it's going to have to be a more generalized therapeutic approach. Okay, I want to talk a little bit about SCA6. This is a fairly common type of dominant ataxia. Counts for somewhere around 1/4 probably of cases. It's fairly common in the United States, but also in Japan. It's localized to chromosome 19. This is one where we know what the gene product actually is. It's a subunit of a calcium channel that's highly expressed in Purkinje cells. It's a little different than the other CAG repeat diseases in that the normal repeat length is relatively short, from 4 to 18, and the pathological repeat length is quite short as well. It's only about 20 to 31. It's a fairly simple cerebellar cortical degeneration for the most part, and the onset can be quite late. But the age of onset and severity of disease don't seem to be linked to the length of the repeat size like it is in some of the other conditions. Interestingly, there are some point mutations that can also occur in this disease that are associated with other neurological diseases, one of which is called episodic ataxia type 2, in which patients under stress will become ataxic for half an hour. Give them too much caffeine, give them a test, they suddenly become ataxic, and it all gets better within like an hour. So that's probably just a channelopathy. This is what the channel looks like. It goes, like many channels, it has a lot of transmembrane abnormalities, or transmembrane motifs. The mutation that causes EA episodic ataxia type 2 is right here. The trinucleotide repeat is way down in the cytoplasmic tail. It's right up the C-terminus. So it's a little hard to imagine how that's going to have a major effect on the function of the protein. So back in the late 90s, this is right when the SCA gene was being identified. Chris Gomez, who was here at the time in the neurology department, got a bunch of patients together, did a very thorough clinical, genetic, physiological testing, imaging, and then we were able to get 4 autopsies from patients with SCA6. So this was really one of the first pathology reports in the literature on the pathology of SCA6. And this is what we saw. We had a couple of sisters who were, one was 87, one was 93 when they died, and they developed ataxia at about age 50. So it wasn't really life-shortening, certainly, but they had pretty severe ataxia by the time of death. And you can see that the cerebellum is quite atrophic, particularly the anterior superior parts. And when we looked histologically, it was very hard to find Purkinje cells in many areas. Here's one cell. There's lots of empty baskets, which you can see better with the silver stain. A few torpedoes indicating that there are a few cells that are still hanging in there. Again, showing the same thing. The deep nuclei were actually quite normal, even though there's loss of the axons from the Purkinje cells. So you see gliosis, but the number of neurons is actually very close to normal, so there really isn't any loss of the deep nuclei. So it's pretty much a primary cerebellar degeneration with some secondary loss of neurons in the inferior olive. So the neurons here are largely missing now, and these are replaced by astrocytes. Chris did some work early on that suggested it may be a channelopathy, and then he transfected the mutant form of the protein into like frog oocytes or something like that, and showed that there was an abnormality of calcium conductance, but whether or not that's really the mechanism, I think is quite unlikely. But he also had some other evidence to show that there was translocation of the C-terminal end with the expansion that goes to the nucleus. So it may have a mechanism much more akin to the other ataxias that are trinucleotide repeat disease. So the thing that we've shown with SCA1 is also true of the other SCA. So the nuclear localization appears to be a very important component of the pathogenesis. Another group of patients that we worked with is the so-called Lincoln ataxia. This is kind of an interesting group because they are actually of the family of Abraham Lincoln. And if you're going to study a family of a famous person, because they always know that they were related to that person, right? So it was very easy to find these kindreds. There were two groups. There was one that was linked to his paternal uncle and one group that was linked to his paternal aunt. And one of them, one family lived in Kentucky and one family lived in Indiana. And they didn't know about the Indiana family until they had started to go down to Kentucky to gather specimens. And there was some article in a local newspaper that somebody from the Indiana family found out about. And so they wrote a letter to Laura Random and said, you know, we're related to Lincoln too, and we've got this problem in our family. So they doubled the size of their linkage group just by sort of a random chance. So this was work that was done by Laura, Larry Scott, and John Day. And they worked on this for a long time. We were able to get a patient who died. We did an exam some years ago now. And unfortunately, I can't show you pictures of it. It was the least photogenic case I've ever had, because when they shipped the brain to me, part of it was frozen, part of it was fixed. And the part that was fixed was next to the frozen part, and the fixed part froze. So there's this huge amounts of ice artifact in it. And so I was able to work with it, but it doesn't photograph well. But what we saw was, again, sort of a primary cerebellar atrophy, very much like SCA6, where it's predominantly Purkinje cell loss. These patients also have a fairly late onset and usually quite a benign course. They can become quite ataxic, but they don't die of the disease. So Laura's group originally mapped this in 1994 to chromosome 11, close to the centromeric region, which I'm not a gene person, but I guess that makes linkage studies much more difficult. I'm not quite sure why that is. But over a period of about a decade or so, and she worked with a couple other groups, one in Germany, one in France, that also had the same linkage. They found a mutation in a structural protein, basically a cytoskeletal protein, beta-3 spectrin. This was work, she had a postdoc in her lab named Yoshio Okeda who did much of this work. And beta-3 spectrin is highly expressed in Purkinje cells, and it's been shown that it stabilizes the glutamate transporter, excitatory amino acid transporter 4. And this is a very important protein because it inactivates glutamate in the synapse. And so if that is not in where it's supposed to be, cells can be overstimulated by glutamate, and that can cause injury to cells. Also, they found abnormalities in one of the glutamate receptors, so that was also the delta 2 glutamate receptor was also somewhat abnormal. And they were able to show that these were abnormal in the autopsy tissue that we had. So luckily, the frozen tissue did some good as well as some harm. So we were able to demonstrate these changes in the autopsy material. So this was somewhat new as a cause of ataxia, because this is a structural protein. But it apparently has an important aspect in terms of putting things where they belong on the membrane of the dendrites. And when that goes awry, it may cause long-term damage to the cells through glutamate toxicity. The last thing I want to talk about is SCA26. This is another one of Chris Gomez's kindreds, which they originally reported now about 10 years ago. This is a family that is of Norwegian ancestry, mostly in northern Minnesota and North Dakota. They develop sort of a standard ataxia, kind of a late onset, usually with gait ataxia early, which is usually a very common presentation. People have difficulty with walking early on. They don't really have a lot of other neurological problems. It seems to be a fairly straightforward cerebellar type of ataxia. And it has a fairly benign, slow course. So we were able to, well, he did some studies by MRI. So we looked at like 11 different patients by MRI. And what he showed was that there was significant cerebellar atrophy, but the pons is pretty much spared, so it's probably not an oligopontocerebellar degeneration. We were able to get 2 autopsies, and there's another one sort of lurking on the wings now from that same family. And again, you can see the atrophy of the cerebellum, particularly sort of in the middle areas here. Again, the same thing. This is from the other patient. And the pons and olives look entirely normal, grossly at least, but very severe cortical atrophy. The molecular layer is very shrunken. Purkinje cells are almost completely absent, so there's just like total wipeout of Purkinje cells. The linkage on this family put it very close to the SCA6 locus, but it really wasn't SCA6. So looking at sort of candidate genes in this area, Chris thought maybe this eukaryotic elongation factor might be a potential target gene. And this is a gene that's really ubiquitously expressed, and a lot is known about its function. involved in translation of proteins, and it's basically essential for life. If it's not working, you're done for. So it's a lethal mutation if it's knocked out. What he found was that there was an amino acid substitution at codon 596, so a proline to histidine mutation that looked like a fairly normal protein. It had sort of the normal localization in the cell. There was no aggregates like you see in the SCAs that have trinucleotide repeats. But based on some homology with the yeast form of this protein, it was noted that when that same mutation occurred at a homologous site, there was a tendency for a -1 frame shift, so that this may be a mechanism. So what's the potential consequences of frame shifting? So you can get altered control of gene products. So this program ribosomal frame shifting, if it can result in destabilization of message, so then the message looks like it's nonsense and gets broken down, so that you may have decreased levels of protein expression. Or if if you have mistranslated proteins, you have problems with conformation of the protein, and the proteins may be broken down due to the proteasomal response. So kind of the model that he came up with is that this mutation makes a protein that apparently works well. I mean, it's stable, it has a function, but there's over the life of the individual with the mutation, there's an increase in frame shifting with gain of toxic function, resulting in slowing of protein synthesis, perhaps mistranslation or loss of this frame shift function. So basically proteostasis is disrupted, accumulation of lots of garbage within cells, and then eventually leading to Purkinje cell death and ataxia. So to just summarize what I've gone over today, I think animal models of ataxia have really had a major impact on our understanding of these diseases. And I think that the fact that we've been able to discover these genes has really advanced the field beyond. I remember when I was a resident, I had several cases of ataxia, and trying to read the literature was just appalling. They were using lots of eponymic terms that they were used very randomly. Some they'd use them for sporadic, use them for hereditary. They meant nothing actually after a while. So the ability to identify these diseases by their genetic component, I think, has really helped us focus on being able to look at actual molecular mechanisms of disease. I think another thing that has been demonstrated with several different models, including the SV40 model as well as the SCA1 model, is that neurodegeneration isn't just cell loss, that there's abnormalities of cell structure that may affect the function of that part of the brain as much or equal to, or perhaps even more sometimes if you have bad wiring, maybe better than no wiring, basically. And then the other aspect is that I think it's important to look at the neuropathological aspects of the human disease in order to, first of all, kind of define what the issues are. If you want to make models, do the models look like the human disease? And then if you can bank tissue, once if you have some ideas about what the molecular mechanisms might be, you can actually look at the human tissue and see, biochemically or whatever, that what you are suspecting may or may not be true. And this was very valuable in the SCA5 case, for instance. So just for some acknowledgments, of course, Harry's lab has been instrumental in much of this. We've had this great collaboration for 25 years. I mentioned some of the people that worked on some of the projects. I talked about more today. So Rod Feddersen worked on the SV40 model. Eric developed the original SCA1 model, and Tao developed the SCA1 model that was responsive to tetracycline, so the sort of conditional model. Lisa Duvik has been a constant in Harry's lab ever since I've been here. She's really kind of the glue that holds the whole place together. A number of other people, I mentioned some, Ivan Clement, Pam Skinner, were involved in some of the other studies early on in the SCA1 mouse model. Huda Zogby has been a longtime collaborator of Harry's. She's down at Baylor. And then in my lab, Wael Yunus, who was with me in the early 90s, and Luann Anderson has been with me for 21 years now. And then Laura's group, Laura and John Day, worked on the SCA5, and then Chris, who was here for many years and then has now become chairman of the department at Chicago. But Chris and I still are collaborating, and I just got an email from him this morning saying, how do I get an autopsy on this SCA26 patient from North Dakota? So. I'm willing to take any questions or comments. Yeah.
Brent, great talk. Thanks for the historical perspective. In the SCA2 cerebrocortical leukodystrophy, like, what's going on there?
Yeah, I mean, it's a completely different disease. It's probably a disease that's involving oligodendrocytes. It's like a demyelinating disease almost.
Strange.
And they have the same genetic abnormality as people who get an olivopontocerebellar degeneration. So it's very mysterious. And why do all these trinucleotide repeat diseases seem to affect cerebellar system, basal ganglia, motor? It's like it's all motor for the most part.
Right.
Something
Who knows?
we really don't know. Yeah.
More to learn.
Yeah, Pat?
From a clinical diagnostic standpoint, you're not going to be getting a brain biopsy necessarily before death. So would you say the average neurologist would be going the genetic route? And do we have a whole panel, for example, that would permit us to pinpoint this?
Yes, there are panels that are available, particularly for the trinucleotide repeat diseases. But of the SCA 1 through 37, the genes have been identified in probably 2/3 of them. So there are ways to look. And then once you identify it in a family, then it becomes much clearer that the family member, that's the disease that family has. But there are a lot of them that have not even been linked yet. So there are a number of cases that could be sporadic but are probably familial, because you just don't have a kindred that you can put together to do linkage studies.
So there are some NGS panels that can be ordered?
That I don't know.
There are some NGS panels that can be ordered, but NGS doesn't work as well.
Yeah, it doesn't work very well at all for those.
So usually, usually those have to be done separately, like Parkinson's is known to by NGS.
Thanks.
John?
Somewhat along the same line, Brent. From a human clinical point of view, can there be confusion between Parkinson's and the spinocerebellar ataxias?
Mostly not, but there are a few exceptions. SCA3 has a very wide phenotypic spectrum, and one of them is that they're primarily Parkinsonian. Actually, SCA3 is kind of an interesting disease in that there was a disease known as Machado-Joseph disease and a disease known as SCA3. And Machado-Joseph had this kind of wide spectrum. The SCA3s were kind of defined by the fact that they mostly had ataxia. When they started looking at the genetics, they both had a mutation in ataxin 3, was a trinucleotide repeat. So they basically are the same disease. So that was one of the problems with the clinical definitions of diseases, was that you could have the same disease and look like something else, or you could have something else and look like. So the phenotype didn't ring very true. So that's why classification was so bad, I think. Yeah.
I understand that in families that have these kind of conditions, that each onset of the next generation happens at a much earlier age.
That's partially true. If it's paternal transmission, there's a tendency for what's known as anticipation, where there's often a jump in the expansion size. And many of these, like these SCA2s who had the infantile onset, the parent that was affected was a father rather than the mother. There's something about meiosis and spermatogenesis that I'm not quite sure if anybody understands it, but it seems like it doesn't typically happen with maternal transmission. But actually, a couple of patients that Chris had, the children were identified prior to the father actually developing the disease. Mark?
So I have a couple of related questions about this. First thing, the knock-in. So you indicated that you can delete at least in SCA1, you can delete the gene in mice are normal. There's no phenotype. Have the knock-in studies ever been done with a single glutamine in place of the repeat glutamine to see are those animals normal?
Actually, the normal gene in mice has like 2 repeats.
It has 2, right? Okay, so the second thing related to the question that was just asked, what is the stability of the repeat numbers over generations? Does it tend to increase or does it?
Well, if it's paternal transmission, it tends to increase. With maternal, it tends to remain relatively stable. In the mice, it stays stable.
All right, and then the thing that's always mystified me, I know that with these
Large
types of repeat diseases, the vast majority are glutamine. Not all of them are, CAG, but why is that?
Because. I don't know, Harry, you have any thoughts on that?
That's the right answer.
Yeah.
People have postulated that part of the instability is due to what's called a stem loop formation, where you loop out one strand and that structure is favored when you have C and G's paired with each other.
So do you see instability in CAG repeat disease in non-coding regions?
Well, the strict answer to your question is no, but there are repeat diseases like Friedreich's ataxia where the repeat is not CAG, but it's in an intron. It's another right, Alexis? I forget what.
Well, myotonic dystrophy is sort of is a non-translated.
In those cases, like in Fragile X, it's thought that you get a shutdown of transcription, so loss of function. In others, where the repeat is in a non-coding region, but still in the RNA, it's the actual RNA itself that's the toxic.
Okay, thank you.